thank you to everybody for attending this session. I think we're in for a very kind of interesting set of 3 complementary talks. And so without further ado, I invite David Bentley from Illumina to talk to us about genome sequencing.
Hey, thank you very much. Thank you very much for the invitation and the opportunity to join this happy throng. The title I should offer no apology for because Gil gave it to me. But I thought about it and thought, okay, it's a good one to stimulate the thoughts a bit. It clearly, the main effort that is focused my area, my groups, generally my collaborators, has been the technical elements of sequencing human genomes, how to do it better, faster, more accurately, more appropriately. But this does give an opportunity to just slightly put some of that into the personal context as well, because ultimately genome sequencing is expected in some way, either potentially or actually, to impact all of us in our lives and in society, as well as the technical challenges that we grapple with on a daily basis. So I accept the challenge of the title. I guess the more general one is personal genomes, personalized medicine, on the basis that in particular I do want to just touch on that interface between moving from sequencing a genome and collecting data from this very pure, very digital information and actually looking at what impact it will have in the future on medicine and the promise, the hope, or indeed the uncertainty of personalizing medicine through knowledge of our genomes. So let me start off with a little bit of the disruptive technology. This is not going to be in detail, it's just to give you a flavor of what we're talking about. But it is important to recognize just in this period of uncertainty, there's an enormous transformation in the ability to generate sequence data. This is just dated since the beginning of the century, and you can see that with a mark here where the human genome was finished, was where the current capacity in terms of billions of bases per instrument per week was quite simply not on the scale for the entire duration of the Human Genome Project. And technology, although it developed hugely in those areas, had not undergone such a radical change. Some of the early promise of the human genome was, should we wait for new technology before we sequence the human genome? Jolly glad they didn't, because the available Sanger chemistry and sequencing methodology took us to a successful, completed human genome. But you can see what's happened since then, since the publication of the human genome. You've seen a massive increase in throughput, really unprecedented, and partly fueled by the increasing availability of good reference information, which the human genome and other reference genomes have provided just as much of an important platform as technology itself for actually bringing about this huge change that we now face. To put a couple of figures on it, roughly a 10,000-fold scale in throughput and a 10 to the power of 7 scale in parallelization of technology. Parallelization has been the main lever here in generating huge amounts more data, from a 96-channel capillary machine, for the sake of argument, to 1.5 billion reads per run. It's just parallelizing to generate more data at the same time. And there may, of course, be other levers in the future. So this has given rise to what we call a tipping point in human genome sequencing, where we see the huge drop in cost of sequencing an individual genome or genomes in general. And you can see placed on this curve, just in the last 3-4 years, the first individual genome from capillary data, the Jim Watson genome from 454 data, marking a change in technology, through to individual genomes just in 2008, and then a little while later, the first 170 appeared in some form or other in GenBank. And clearly, as the costs drop precipitously, you do see this potential to increase massively the number of genomes that can be sequenced. And we really have reached this point in the last 12 months, perhaps, of considering that individual genomes can be sequenced, both in research and ultimately in the personal space. And I'd like to touch a little bit on both of those by way of background. In terms of making it a reality, that does mean that once we've reached this concept of being able to do 300 gigabases in a week, that does mean on one instrument we can do one human genome in 3 days. I'll say a little bit more about what it means by a human genome and what it does not mean. No human genome, including the reference, is complete, and these are certainly not complete by every base in an individual's genetic code, but they are a very comprehensive representation of the human genome. For 3 days, today, I guess, down to $10,000 per genome, which really puts it in the context of medical costs and other budgets and research budgets. and rapidly continuing the trend down towards the $1,000 genome for direct costs, and I'll discuss a little bit about what the other costs might be that are excluded from this figure. But clearly that trend is set to continue. The curve has not bottomed out, and we do look for a greater coincidence of the cost and ability to produce a high-quality human genome and to put it in the context of interpreting it, ultimately moving from just research to personal and diagnostic use. Just to go back in history, all of 2 years, this was our first experience of looking at an individual genome and looking at how complete or how accurate it was or was not. We actually determined an anonymous genome, the African genome, from a HapMap sample, 3 billion bases, to a depth of around 40x on average, and determined that overall the coverage of the reference sequence was around 99.9%, depending on the method you used to analyze it. This was the Sanger analysis using MAC. And the consensus accuracy, it's a big figure, but of course you expect that if you're going to a depth of 40x, you're ascertaining on average every position 40 times over, you expect a high level of consensus to emerge from that data. And I'm going to come back to the principle of that in a little bit more detail later. Exposing the fact that at least 10%, however, of the reference genome cannot be analyzed for variation, 90% can, but not a technology-specific problem, but some of the genome is just that repetitive that you really cannot resolve exactly where to place the reads in a resequencing experiment. So you are left with interpreting 90% of the genome at the present, or matched to this particular technology. But nevertheless, the majority of genes and exons and features of biological relevance are well captured in this product, even if it is not complete. And it gives one the opportunity then to map to the known public annotation of gene structures and functional elements and regulators and so on, the possibility to look for variation that is attached to this particular individual compared to the reference. And this particular analysis revealed 4 million variants or so, SNPs, indels, some structural variants, to varying degrees of certainty. And in particular, just from the single nucleotide polymorphisms themselves, a quarter of them were novel. And that already places a burden both of proof that they are real calls and also indeed a burden of interpreting these novel variants for which there is no previous knowledge or previous history. To illustrate the importance of putting human genomes more into the, by way of being a one-stop shop or a single source of information, it's important not only to detect where a sequence, where a genome differs from a previously known reference, but also can you actually characterize fully the variation that is novel in the genome. This particular example therefore illustrates where the resequencing mode leaves off. The blue reads here are aligned to the reference essentially perfectly. You can place them uniquely against a previous reference, so you know where they stand in this new individual, and you can compare the two to look for differences. And the same is true here. But here you see in the middle a portion where actually there are reads which did not align to the reference, but are actually later retrieved from the unaligned reads and actually added to the assembly, so that you eventually get a contiguous representation of a novel variant, in this case a deletion in the sample compared to the reference that was previously known. So there's greater challenges in terms of not just aligning to what we already know, but to assembling something de novo and describing, decoding the novel aspects of any particular genome. Can to varying degrees be quite complex and is an area for ongoing research for years to come. I'm not going to go into much more technical detail on this score, but instead just to reflect for a moment on bringing in the personal aspect, which actually this surprised me. This was the front cover of Nature, which actually featured when this genome was published, and actually chose to take the personal angle. Here's the front cover of the original human genome public sequence, safely contained within the safety box. Break glass when ready. How will we tell when the genome is really ready for prime time or ready for personalization or personal use? But when you do let it out, your life will be in your hands in a very new and personal way. And I think it's perfectly fair to say that in the 2 years since this appeared, clearly we are seeing a great deal of focus on the personal and social change which accompanies the ability to characterize whole genomes to the extent that we saw just then. Somebody's got a message. It's not me. This is perhaps the most technical element of The challenge being, if for the sake of argument, to take an individual, to sequence the genome, and to characterize the genetic variation which represents that individual's inheritance and ultimately how that individual in ways interacts with its environment. And so there are just 5 classifications here. Just to give you the technical detail of the current format of data for the Illumina platform in particular, which is not true of all the other platforms, but it illustrates here is the reference sequence in the black line, and what we typically do is take short fragments and we sequence from either end, and you get a pair of reads. And there's the actual sequence information in each read on either end, but there's also the information of the spacing between the 2 reads as it is mapped back to the previous knowledge. From that information, there are various methods to characterize different types of genetic variation. The single base substitutions emerge essentially quite logically from aligning and looking for differences. Short indels actually require a different methodology for alignment and benefit in particular from gapped alignment. So the initial alignment will simply record a poor aligned read, which might be rejected in simple formats, but by introduction of 1 or more gaps, you then increase the accuracy of the alignment, and as a result you record the fact that you have got a model that fits a consensus of a single base indel in this case. Through to the larger structural variants which demand a different aspect of the data in current format. If we have the existing structure which looks like a normal paired read aligning to the reference, if however you actually observe multiple events when the reads appear to be further separated than you would expect, it's not because the DNA is larger, because you know what you put into the experiment. It is because there is a fundamental structural difference between the reference and the new sample, and in that way you can look at more gross rearrangements. And finally, copy number variation, and also by looking at the orientations of reads, altered orientation means a different type, an inversion in this case, as opposed to a deletion or an insertion. And finally, of course, copy number changes, where reads actually pile up because they're not resolved into adjacent sections of a tandem duplication, for example, but you see them as signatures nevertheless of deeper read alignment, which is reflective not of that's the actual structure, but the structure is one of there is more than one copy of this sequence, and the sequences have collapsed in the alignment. But nevertheless, it gives an indication of a copy number variation. So all these different types of variation already involve different types of analyses of area of extremely active research and improvement in the accuracy of these calling algorithms and the ability to display and to validate indeed the results of these. But together, you're looking at capturing this information to actually describe a single genome. I just wanted to capture this moving on from being able to collect information from individual genomes to a greater or lesser extent. This rather nice summary that was provided by Les Biesiger last year, 2009, yes, to illustrate how such a study fits into the context of other studies. So this is a conceptual slide. If you look, there are 3 axes starting from the origin in the middle. There is genome breadth in terms of the amount of genome that you cover from a single gene to a whole genome. There are subjects, the number of cases that might be included in a comparative study from one single genome to lots of individuals, thousands of individuals, perhaps in a clinical study. And also the amount of information that you actually have available on a particular genome, the amount of clinical information. The ideal study, therefore, being that you could actually generate complete information on a large number of genomes or subjects for which there's a great deal of clinical data available. And that would be a really archetypal study from which to examine a great deal, to correlate all that genetic variation, which I was talking about on the previous slide, with phenotypic information from the individual. Here you see matched to it individual genomes. You get complete genome breadth, you get almost no subjects at the moment, and a little bit of clinical data perhaps if you're lucky. Single gene studies, almost no genome breadth, but you can do them on quite a lot of subjects, and you can get a variable amount of clinical information depending on study design. And the 1,000 Genomes project, which I know Gil and others have been very involved in, gives you great genome breadth on quite a lot of samples, but at the moment these samples are anonymous and don't have any clinical information. So you can see how different components are invested in different groups, and ideally you really want to maximize all 3. So individual genome sequencing has one part to play in this, but it fits in the context of applicability to many individuals and individuals where there is clinical information or other phenotypic information available to actually maximize the value and the ability to interpret the genetic variant that you found on the previous slide. So this is going to introduce, and I'm going to touch on the challenge of interpreting individual genome sequences for a moment. and also the convergence perhaps of individuals, consumers, as it were, the well, hopefully us in this room, and also clinical cases, actually from medical situations where genome sequencing might be of interest or value in the future. So let me talk a bit about personal genome sequencing, and I wanted to go back to an old slide, which indeed several of you in this room actually probably helped me conceive and draw and advise. But it's the illustration of how one sees personal genome sequencing information emerging and making maximum use of public domain information. So here is public domain information, broadly speaking. It's a well annotated reference genome. It has a gene structure. It has features like an alternative promoter. It's got functional variants in it, which are known to be effective changes, genetic variants which affect function. There are perhaps some well-defined regulatory elements, and there's a position in the genome, all-important position in the reference. And this comes from a combination of many sources of information, a massive and growing resource of information. But to put this into a clinical context, of course, is almost impossible. We can't have a doctor look at the entire composition of public knowledge about genomes. And instead, what is needed, indeed what is currently not really available today, is the opportunity to have a filtered out, very small set, for example, of risk variants. And I've said here approved for clinical use, because there is a level of approval and validation which is very important in medical genetics. But to illustrate that from this massive information, you can filter out a small set of variants which really would be of high value in interpreting risk or diagnosing a particular disease. And this simple lookup table, indeed, is something that could be regularly updated as the public domain, as research increases its understanding of a particular disease. But the actual table would remain small, well-informed, approved, and continually updated. It's a bioinformatic tool by which you can keep track of public information in a manageable way. On the other side, you have an individual genome sequence, preferably perhaps owned by the individual, and these two resources come together in the basis of a consultation with consent, such that the personal information can be interpreted on the basis of public information, and that selected risk information might be used to help inform a clinical decision. So this is a goal which has really been pursued by many for some time, and I just want to touch for a moment on some elements of the workflow. I'm not going to get so much into the ethical discussions here, but the importance here is that we do in fact have a workflow by which we offer genome sequences to individuals. And one of the key features of this process is that it's actually physician-led, in contrast to the recent debates about direct-to-consumer. Should it be allowed? Should it be regulated? This is already regulated on the basis that a physician takes part as the gateway between the individual and the genome sequence which is provided to them. So the doctor orders the sequence is provided back to the doctor, and it is the doctor who then delivers the information back to the patient. Maintains confidentiality. It's a highly regulated, clear lab, CAP-approved lab, and goes through all the necessary QC processes as far as indeed as they are defined by the regulatory authorities. So what about the data that comes out? And I said I'd return to the concept of consensus accuracy for a moment. And clearly there's been a lot of question, well, is next generation sequencing or current generation sequencing really going to provide information of sufficiently high quality for doctors to use it in genetic interpretations? And here is the question that people are looking at. Here on the left-hand side is what is currently accepted as the gold standard for diagnostic testing. It is an amplicon, which selects a particular segment of a gene which may be of interest. And the sequence of the amplicon may be determined on both strands. And you see here a peak corresponding to a particular base, and that's a clear signal, a G, a C. And you can see one peak nicely corresponds to one base and matches the reference sequence. And you move to this point here, for example, where actually there's a drop in quality, and there are two different signals being reported for the one position. Now, throughout the entire period of the Human Genome Project, of course, such a base would have been declared low quality, and we would have looked for an alternative, unambiguous call of a particular position in the genome in order to create a reference. So this is now what the reference genome would have concluded was a low quality base, unable to draw a conclusion from. That was easy because every segment of the reference was only one haploid sequence, whereas here we are genuinely dealing with a clinical sample where there might be a heterozygous, there might be two right answers for a particular position. But it's perhaps interesting just to reflect for a moment that the gold standard in genetic testing at the moment is a base that would have been rejected from the Human Genome Project dataset. Moving on to the next generation sequencing platform here, you have the same segment, if you will, being sampled from short reads in the method I described earlier, and indeed any method seeks or most methods seek to go quite deep. And you see here many multiple independent determinations of this particular region of the genome, and I've highlighted, as it were, conceptually the same position that we saw as the heterozygous position here. But here, each read here comes from ultimately a single molecule, which in this case has been amplified. Some systems do not amplify the segment, but they determine the sequence of a DNA molecule directly. And because it's a single molecule, it has to have one or other of the alleles, depending on which chromosome it came from. And so what you see here is multiple independent high-quality base calls capturing much more the original concept of the human genome sequence. And therefore, each one is a right and high-quality answer, and indeed has a quality metric, or can have a quality metric attached to it to reflect on the high quality. So essentially this, perhaps just to return to the question, should I sequence my genome now? I'd actually be much happier with a diagnosis based on this information than I would based on this information on the concepts of which I've just described to you. So one answer is yes. So let's move on to interpretation. And essentially I should say that the same principle, of course, can apply to any position in the genome sequence, subject to challenges of difficulties of alignment and repeat sequences, as I mentioned, or areas where perhaps you don't get the coverage to really get that kind of depth. It is possible to impose quality metrics or assess metrics to provide metrics to assess the quality of the ascertainment of any call at any position. So again, if you don't have a good call, at least you know you don't have a good call and you shouldn't actually interpret it. But let's go on to the thorny business of interpretation, which I alluded to in Les Biesiker's ideal study model. The idea here was to go on from our initial process of sequencing genomes to actually indeed provide the physician with some information based on the model that I described earlier to guide the physician in terms of what he might or might not need to say to an individual. And a doctor does have a duty of care to pass on information that is of obvious potential risk, high risk, to the individual. So the idea then being to annotate the individual genome sequence, at least at the well-known mutation sites, in particular to assess the risks at those sites, and to prepare a report that perhaps just focuses on those very high-risk variants. Say for the sake of argument, an individual has their genome sequenced, they get told they're perfectly healthy, and 3 years later they come back and they've got Huntington's disease. And they can say to the doctor, surely you could have told me that, I thought that was a well-known mutation. And the answer is yes. So we're trying to avoid that sort of situation. We're not trying to report on whether you have a 1.157 increased risk of diabetes or not. That's a whole different story, and I'm not going to get into that. So we focused on the high-risk variants and asked, well, how could we find out? The sources we used, HGMD, a well-known published source of published gene lesions responsible for human inherited disease. So these are all the Mendelian disorders, for example, Duchenne, hemophilia, Huntington's indeed, and a number of other genes where there's a very well association with a high risk variant, of which there are 97,000 entries, although only 53,000 are actually mapped to the genome even today. Some of this goes back 20 years. It precedes genome sequence and genome coordinates, and for that reason not all of them have been mapped yet. There's also a well-known public database for pharmacological variants, pharmacologically important variants. Again, 11,000 entries, but only 1,000 have SNP and drug information available. And then we thought we should also take the genes which are previously implicated in the HGMD database, some 2,800 genes, and also look for novel mutations that might nevertheless be indicative of a high-risk variation in the individual. And so we did this just in a pilot genome. We discovered in the, I've already mentioned one piece of bad news, if you will, that of the 97,000 entries, only 53,000 can be mapped. Well, 99.3% of them were in fact covered in our consensus sequence, so that's pretty good, but not 100% ascertainment. And initially, you see a very small number of these variants actually were called in this particular genome, but there were 16 homozygous disease-causing variants in this genome, and 48 heterozygous potentially disease-causing variants in this genome. And they actually, looking at the homozygous disease-causing variants, a whole range of conditions, von Willebrand disease, Wilson's disease, adrenal hyperplasia, G6PD-H deficiency, all of which, if you think about it or if you ask a clinician, would conclude that death in early infancy is most likely in the case of this particular individual. So we didn't believe this, and so we went on, had a look at each of the 16, and we discovered that for one reason or another, every single one of them could actually be excluded, either on the basis of errors or subsequent information. It emerged that a variant was common in the population. So some of it is quite genuine evolution of knowledge over the time. Some of it is errors, and some of it is poor interpretation. But after that, none of the mutations remained a consideration. And essentially the same exercise we repeated over much more of the study, and eventually decided that the genome sequence of this individual was in fact clearly compatible with life, which was just as well because he was a rather well-known chief executive of a well-known biotech company. And he did give permission to make that knowledge available. But that is actually one of the hard points. Having illustrated to you the difficulties we face in moving from quite highly developed understanding of DNA sequence to interpreting it and looking at the sources of information we have available, and then trying to make judgments which actually affect people's lives, is by no means simple, and is plagued with a great deal of different levels of difficulty, which require a whole load more information, research, and accumulation and accessibility of information. And I should make one more point as well, because there has been, and I will allude to it at the end of this talk, there's been a suggestion that if you're going to sequence somebody's genome, it's of maximum value if you do it very early in life, so that you can capture the possibility of risk or the ability to prevent diseases throughout life. The reason we knew, of course, that those mutations were mostly wrongly interpreted is because the individual was already 55 years old. But if we had actually done a newborn in this case, we wouldn't have been able to do the process which I've just done and to actually debug the system. So it's an important point. Perhaps I ask again, should I sequence my genome now, or should a newborn have their genome sequenced now? There is some question about the value that you would get, the veracity of the message that you would get. Moving to a more compelling medical situation for a moment. So this has so far been about sequencing the well and asking, perhaps in a preventative medicine sense or a diagnostic sense for the future, what might happen. Here we're looking for a moment at a really contrasting end of the spectrum of a really important disease, and of course cancer is a disease of the genome. These are somatic changes largely, but in combination with germline variants inherited as well, which together perform or comprise a combination which eventually can trigger tumorigenesis and indeed the further evolution. I flag for a moment, first of all, a little bit of history, again our own first experience of this in collaboration once again with Sanger and one or two other groups, to characterize a tumor normal pair. This is now becoming fairly commonplace in the literature, so I don't need to dwell on the concept in any great detail. But to illustrate the point, this is a range of different substitution, or I should say, yes, somatic substitutions, copy number variations, inter and intra-chromosomal rearrangements, which together have been discovered to be present in the tumor, absent from the normal by a high stringency filter. And so a whole range of mutations, somatic mutations, which in some way or other reflect the fact that this individual is indeed a melanoma case, and some of these variants are passenger mutations, of course, and others actually in some way were causal or drove the cancer. And indeed the spectrum of mutations does have a signature which reflects the exposure to ultraviolet light, sunlight, which actually was the chief mutagen in the first place. But the concept being, first of all, you can then characterize an individual tumor, take a snapshot of this tumor at a particular stage in its evolution. But of course that is not the whole story about cancer, and this study taken from the recent publication from the WashU group from Li Ding et al. Illustrates how they went on to look at a single cancer in multiple stages, the primary and the metastasis, and also indeed I think a xenograft as well. So they took a bit of the tumor out and planted it into a mouse and then looked at the genetic mutation profile in all of those different samples, primary, secondary and xenograft. And what you see here across the bottom is a list of particular genes which were mutated, and a list of mutations in the a list of mutations in the tumor, which you can just see they're ranked from left to right by the percentage of mutations in the primary tumor, So this gene is highly mutated in the primary, this gene is not at all. And then in green, the mutations in a particular gene which are associated with the metastasis. The point to see from the distance that some of you are at is quite simply that this profile is very different from this profile. And so the accumulation of mutations in the primary tumor is one thing, and somewhere in there are the mutations that actually were the drivers giving rise to the tumor. But here are additional mutations which may be responsible for the further evolution of the cancer following the initial diagnosis, and indeed these are in the metastasis. So perhaps some mutations here are responsible or are signatures of metastasis which were absent from the mutations in the primary tumor. So you've gone from taking one snapshot of a tumor to taking multiple snapshots which reflect different stages in the evolution of the cancer. I haven't really got into it, but I think I have a little scheme here, yes, which illustrates, just to acknowledge one of the number of groups, in particular Navin et al., where they've actually gone to look at not just the overall representation of a particular mutation, but actually going down to look at single cells, to look at some cells have different mutational spectra from others. And of course this is a very much a prominent area of increased understanding at the moment and research, to understand that cancers are not only evolving in this sort of mass analysis, but they are evolving in very real terms as single cells within the tumor. There's a great deal of heterogeneity, and some cells have different genetic characteristics from others, and therefore are at different stages of evolution. And a minor component today may become a major component in the tumor tomorrow, and this may be part of the whole cancer evolution. So the concept of cancer evolution is much more than a single snapshot of a tumor, but clearly we are in a position to begin to characterize both the evolution and the heterogeneity of tumors in greater detail, and this drives us towards ever more sensitive and high throughput methods for sequencing. To return for a moment to the medical angle, I still quote this example, though it's 18 months old, a talk from Mark O'Mara at British Columbia, where he cited a study in which he looked at a metastasis in initially a tongue cancer. These were now secondary cancers in the lung, and he actually took the approach that we've just been talking to. The individual was put on a particular treatment, ilotinib, chosen as much as any other drug could do potentially of value, but the tumors were unresponsive, the tumors did not grow. As a result, they then went on and sequenced the entire genome and transcriptome and looked at the biological components that are known to be involved or implicated in proliferation. And they found no less than 17 changes in one particular regulatory signaling pathway, and including a particular target which was amenable to an already approved drug. And they actually, in fact, they also found that the target for the existing drug was mutated and therefore would not respond in any case. So they could have actually, if they'd had the sequence information, this first round of treatment, which was expensive, would have been avoided. And a more appropriate tailored treatment would have been indeed successful, because the switch to the drug recommended by the information gained from the sequencing indeed was actually successful, and the tumor was certainly reduced. So just one example of using whole genome sequencing, potentially the concept of capturing all the information in a hypothesis-free way to actually extract information which is a potential clinical use. And I've gone from one end of the spectrum rapidly to the other. So just to wrap up, what's next? A few slides to look ahead in fairly simple terms, if you will, to perhaps promote discussion or questions. And to recap, we are today actually sequencing a genome for about $10,000. We do see it coming out to further improvements in throughput in the short term will both increase the speed of turnaround. Turnaround is also very important in clinical situations if you do want an answer fast. It's already a big leap to say you want a whole genome sequence in 24 hours, but let's just look ahead to the possibilities, because there are some compelling technical reasons why whole genome sequencing is actually a very straightforward way of analyzing a genetic profile. But we will then potentially see the process accelerating up to a day, and also further reducing in cost to less. And I've said, for the sake of argument, this rather iconic figure of $1,000, which again we can debate what that includes and what that does not include. And where the data might be stored. In particular, there's much talk of a central solution, a cloud solution, or whether it's a local solution Originally, people thought perhaps I'll store my genome on a credit card, but people tend to lose their credit cards every so often, so that's not very encouraging. But also, of course, there are vehicles either with local storage possibilities. You can store our CEO's genome on his iPad, and he's got it on his iPad, in fact. But you can also use these devices for remote access to the central locations. I think I'm not familiar with this, and there are many in the audience who know more about this than I do, but I just did a little bit of fact-finding on the question of the cost and ability to store information from whole genome, indeed the ability to analyze it. And so just to look at the computational challenge from a light-hearted point of view. Computational challenge might be recorded on a scale of 0 to 1, for the sake of argument in units of H, where 1H is, for example, 10,000 cores and 10 petabytes. Of course, this could be seen as a real prima Rolls-Royce kind of facility. And of course, there is the potential to go further into the clouds in the cloud computing facility. In case you're wondering what the unit of measure of computation is, it is, of course, the Hingston. So this is the current capacity of the Hingston site. And to put into context what we're currently using, this is the entire Chesterford site, which analyzes multiple genomes in a week, and does illustrate the possibility within the particular CLIA lab that I mentioned that's currently offering whole genome sequencing to individuals, including the process, is actually working with about 0.02 Hingstons, so significantly less than the entire Chesterford facility. And from a review from somebody, I think from Cold Spring Harbor, again, it's only one data point, so I look to you for further information. But the suggestion being that you can actually access this kind of resource required to analyze a human genome from end to end is actually available on a cloud for a relatively modest cost. And I look for validation of that information from this audience or future audiences. But certainly the argument from some time ago was that actually the cost of a genome does not include the cost of analysis or interpretation, but at least computationally it does look as if a great deal of development elsewhere in the world is actually helping to make the possibility of analyzing the data once it's been generated more of a reality. That does nothing, of course, to touch on the challenges of interpretation. So if I was to sequence my genome now, could it be stored? Could I access it? The answer is probably yes. Looking at applications, I've mentioned cancer diagnostics. We've seen on the one hand the ability to capture a whole cancer genome at one point in time and actually conclude something of value, something which actually does change treatment and actually brings better use of existing approved drugs as well. This is not waiting for a new cure. This is making better use, giving the clinician better information with which to choose the more appropriate drug than was previously used. And the cost of each one of these treatments is actually greater than the cost of sequencing the genome today. So the cost benefit equation really does already add up in this situation. I've alluded to newborn screening on the basis that I think I've already mentioned, the idea being that if you're going to have an individual's germline sequence it is most appropriate to be able to use it as early as possible in life. Indeed, a certain level of threat and mortality does happen very early in life. We've had a number of requests for newborns where they simply cannot diagnose what is wrong, but it's something serious, perhaps an immunodeficiency where there may be already 60 genes that could be implicated in the immunodeficient phenotype. Sequencing the 60 genes is already a lot more difficult than sequencing the genome and bioinformatically selecting out the 60 genes. But clearly there are questions of interpretation around that which I highlighted. Looking a little bit more at some of the issues, I've already mentioned interpretation at some length, but if you focus the question on validated variants, and the question of clinical utility of variants is a long-standing debate and will continue for years. Interpretation has to be very carefully handled. It's in its infancy. Inevitably, the genome sequencing world itself has had a 10-year head start on interpretation, really, and so now the challenge is amongst us. Integration, particularly into, this is not health and safety, this is healthcare and society. There is an important element of integration in terms of the ability to actually see how this kind of data would integrate into medical practice, what the social implications are, privacy, confidentiality, all the usual arguments and perhaps some new ones as well. The question of regulation This is kind of information that the regulatory authorities have not really worked out how to regulate, but there is an important element and an ongoing dialogue, certainly between all of us data providers and technology providers and the regulatory bodies. But there's a whole area that will need to be resolved there. And the question of whether people want it anyway, whether it really is valuable, whether there really is uptake at the end of the day, is a debate, or indeed whether this technology gathers information which then goes for a more targeted approach to clinical intervention in the future. So that's what's next. I just finish on, as always, if in doubt, look to the national dailies, and the Times over here actually published this report, which very interestingly, it's a year ago. All babies will have gene code mapped at birth within 10 years. This came out February last year. And remarkably, I think if this article had come out 2 years earlier, there would be an absolute hue and cry, I think, in the public media. But actually, it was received cautiously with positive sentiments in the follow-up correspondence and articles. So that was a very interesting change, rapid, I think, measure of change in social awareness and understanding and indeed acceptance. And reinforced indeed just a month and a half ago, once again. In fact, this is Francis Collins, now head of the NIH. Of course, has been one of the many pioneers in the human genome world, looking again to continue to promote the concept, very much, I think, in a sensible and safe way, but to look at the prospects in the future of DNA sequencing. Well, and he does really mean whole genome in terms of the article. And if you do read these articles, I think there's a tremendous amount in here. These are done by Mark Henderson, who is one of the best, if not the best, journalist, certainly on this side of the Atlantic, covering these areas. He really hits on a lot of points. So we look to the Times to tell us what the next step will be. Thank you very much.